Okay, let's go ahead and get started. A couple of announcements. First one is that the guest lecturer on Friday is going to be Dr. Michael Bartlett. Some of you will have had him for Principles of Biology, at least the first term. So he'll be talking about chromatin modifications, particularly histone modifications. I'll be giving a PhD student in Copenhagen, Denmark, a hard time on that day. So second announcement has to do with our clicker thingies. Turns out because Macmillan wants to make more money off of you, they have this whole reef clicker and, et cetera, new stuff, which is, I think, totally worthless. And so that's why we're sticking with an internal system with old clickers. Old clickers work just fine. But what that means is, unfortunately, the online registration at their website doesn't work. So at least it doesn't coordinate with our D2L system unless we pay them ridiculously large amounts of money, which we're not going to, because it would unfortunately then come from you. So that means that to register your clickers, you need to go to D2L and answer the quiz, which is under activities. About 60 of you have already done that. The rest of you, please go ahead and do that. All that you need to do is enter your clicker number. And as long as you enter that clicker number, you'll get a point. Hopefully that clicker number is right. There's no way for me to actually check this. So be very, very sure that that number is correct. So are there any questions about that whole clicker business? I had a couple people right before class asking me about it. Your clicker responses will be recorded with that clicker number. The key in terms of registration of the clickers is making sure that that gets connected to you, as opposed to just a clicker. So questions, comments, worries about clickers. The 6th edition of the textbook should be on reserve in the library. Yes? Anyone look for it? No? We haven't tried yet? It's not that early in the morning. At least I don't think so. OK, so speaking of clickers, let's move on with it. Our first clicker question for today is alpha helices and beta pleated sheets are similar in that they both repeat every 3.4 amino acids. Our primary structures have hydrogen bonds between amino acids that are less than 4 amino acids apart, have side chains that project away from the peptide backbone, or motifs. Again, feel free to talk about this. Yeah.
This one your quiz says on the back. Is it the IC number or just this one at the bottom?
That one at the bottom.
Okay. There's 4 sets of numbers.
Yeah. 30. Ten, 5, 's pretty amazing. When I say 10, about 10 people voted exactly at that particular time point. With clickers, you can change as many times as you like. It's just the last one, which is actually recorded in terms of what that is. So probably the best thing to do is when I put up a clicker question, just hit a button, and then we can go back and take a look at it. So what do you think? Most of you thought side chains that project away from the peptide backbone. Nobody liked repeat every 3.4. Why not? It's only alpha helices, exactly. Our primary structures? No, they're not primary structures. They're secondary structures, exactly. Have hydrogen bonds between amino acids that are less than 4 amino acids apart. Again, this is alpha helices, not beta sheets. are motifs. What's a motif? Motif is not really a tertiary structure. Motifs are things you see in primary structure. So they're going to be particular amino acids, such as leucines every 7 amino acids. That would be a motif. And we'll talk about zinc fingers later on as well. These are specific amino acids that are specific positions in the primary sequence. But of course, yes, most of you are right, maybe because I said it properly, or maybe you knew it already, that it's side chains that project away from the peptide backbone, either in terms of sticking out for alpha helices all the way around, or up and down if you're talking about beta-pleated sheets. So our next clicker question is, the largest protein family in most vertebrates is that of protein kinases. This means that vertebrates probably have very few proteins, regulate many of their proteins by covalent modification, had many genome duplications, have most of their proteins that are very similar to each other in sequence, or regulate their metabolism by phosphorylation. Again, feel free to discuss. Ten, 5, up to 107. Okay. So what do people think? They mostly think that means they regulate their metabolism by phosphorylation. Unfortunately, I don't agree with you. So let's go through some of the other options. And unfortunately it overlaps here. That doesn't work terribly well, but at least I've got it on my screen here. So have very few proteins. Nobody liked that. I think that's pretty reasonable. Regulate many of their proteins by covalent modification. What do kinases do? Kinases do what? You don't have a clicker question. I don't know who you are. So what do? They put phosphates onto proteins. Sometimes onto DNA, but we haven't talked about that yet. How do they put them on there? Through a covalent bond, right? So it's a covalent modification that happens. How is that different from G proteins? G proteins have a GTP molecule that they hydrolyze, but there's no covalent bond to the protein for GTPs, with the other major switches that we talked about. Had many genome duplications. What would that do in terms of making protein families? Actually get quite a few of them, but it's a whole genome duplication, not a gene duplication. So you don't get quite as many. If it had been gene duplication here, that actually would have been a pretty good answer. And have most of their proteins that are very similar to each other in sequence. That's the beginnings of a protein family, and we talked about that last time. But they don't necessarily have to be similar to each other in sequence. At least not detectably similar to each other in sequence. What do they have to be similar to each other? How do they have to be similar? Their structures have to be similar to each other, which may or may not mean that they've got a similar sequence at all. And then regulate their metabolism by phosphorylation, which is what most people thought. But that's probably because they thought phosphorylation and said, hey, it's kinases. It has to be phosphorylation, right? Sound like a good description? But metabolism for vertebrates is basically identical to metabolism for almost all other organisms. So why would vertebrates themselves have particularly large numbers of protein kinases? Could be, but I think a better answer and again, I just think this, is B rather than E, because metabolism is a much more general kind of process rather than something which is going to be very specific for vertebrates. What this means is that it's probably not a very good question, which gets back to how I grade my exams. I always set the high score to 100%. High scores on my exams are usually about 40 out of 50. So that means that 10 points basically disappear. So this would probably be one of those points that more or less would disappear. And that is to your advantage because you get to pick of those 10 the ones that you missed, as opposed to the ones that everybody else missed. That's what the advantage of normalizing to the high score is. So that also means if you have questions about more than 10 questions on an exam, come find me in office hours. If you have less than 10, I'm not going to be very sympathetic. Okay, so that's okay. B is the correct answer. Good, I can hide this. With any luck, this will be appropriately recorded. I will try and remember to save it as opposed to last time when the new software doesn't save it the same way. There was one question about clicker questions from last time, whether I could put the answers up there. I'll try and do that for all the clicker questions before the midterm as well. Yeah? It's under activities. It's a really kind of funky way D2L is set up. So if you have activities, which is the main menu at the top, and then activities pulled down, there's a quizzes under that. Okay. Yes?
I have a question about your GTPase.
Yeah.
You kind of finished that last time. You said that it acts as a timer when it's hydrolysis.
Yes.
What determines that length of time?
What determines the length of time of the timer on a GTPase for the GTP protein? That's a great question, and it's mostly going to be covered in cell biology next term. Has to do with the other proteins that are involved in interacting with it and also the specific G protein itself. And so you can modulate that timing quite a bit based on other partners. Yeah? Wanted to finish up talking about the last couple of proteins and protein modifications. We talked about G proteins there for just a second. The other major signaling proteins that we're going to talk about here have to do with a different kind of covalent modification, and that's the covalent modification due to ubiquitin. Now, ubiquitin is a small protein. You sort of see the little question mark here, which is supposed to represent ubiquitin. Not surprisingly, you find it in all kinds of different organisms. That's why it's called ubiquitin. And that sequence is very well conserved. What happens with ubiquitin is it's a about 70 amino acid protein, single domain, that can attach to lysines on your protein, and actually really any free NH group, but most of the time it's going to be lysines. And that gets put onto proteins, again, as a signaling molecule. In many cases, that signaling is going to be for degradation, which is polyubiquitination. And that polyubiquitination leads to degradation by the proteasome, the cellular garbage disposal. But it turns out ubiquitination can be used for a whole bunch of other things as well. So how do you put ubiquitin on? It's not quite as straightforward as just having a kinase, which will put on one phosphate. Here, you have this whole group of proteins called, really conveniently, again, the molecular biologists have a huge amount of sense of humor here, E1, E2, and E3. Eek. Just a horrible nomenclature, but the E1 protein basically takes ubiquitin, hooks it onto itself, and this is that high-energy intermediate that we talked about before. We talked about some of the enzymes that are involved in, basically, for the most part, as far as this course is concerned, making polymerization. So first, this binds to ubiquitin. That gets transferred to the E2 protein, which is also now a high-energy bond. This gets bound to the specificity protein. This is called the E3 protein or ubiquitin ligase. So E1 and E2 are very nonspecific. They're going to pull ubiquitin out of the solution, hook it up to E1, transfer that to E2, And then when E3 binds, now you'll have a good amino side chain. Again, most of the time this is going to be lysine. Sometimes it's the N-terminus. Sometimes it's going to be an arginine. But then you add a ubiquitin to this. That'll serve as, just like the case for phosphorylation, you have a big group on the outside which has a particular charge. It's going to change your structure. Here, you've added basically a whole new domain to your protein, which again, not surprisingly, is going to change the structure. And so that's all that's going on there. A little higher resolution view of what this looks like. These are the actual proteins involved. This is the E3 protein, the so-called Cullen protein. One of the reasons I want to mention this in more detail, this is the particular protein, in fact, one of the proteins in a protein family. Not surprisingly, these are vertebrates as well. It's been very intensively studied by Dr. Singer here at PSU. So he works on these ubiquitin ligases and particularly these Cullen proteins. They then here are going to interact with these E2 proteins. It's that Cullen protein which is really going to be the one which gives you the specificity. We know quite a lot about the structure of these proteins now and how they're interacting. What exactly the signaling leads to is still a very active area of research. So these proteins, again, it's the E3 protein, which is a specificity protein, which means you're going to have lots of different E3s. You'll have just a couple of E1s and E2s because they're going to be interacting with exactly the same thing. And then the E3s are all going to be different depending on whatever their target is. And so those differences in the structure, of course, going to lead to differences in function. What are the functions of these proteins? They're basically just binding two proteins together. And so it's that interaction of the two proteins which leads to their activity. And what people have been doing more recently is just looking at interactions of different proteins with each other in order to try and get an idea what their activities are. Turns out it's actually pretty easy to figure out what protein interacts with another protein. The problem is a bunch of proteins interact with a bunch of other proteins. And by the time you try and look at all the proteins, this is the case for Saccharomyces cerevisiae, a relatively simple organism, and a simple set of protein interactions ends up looking like this, just a huge god-awful mess. So to really try and tease apart what's going on here, you either need a degree in computer science or you try and simplify things a lot. And so now this is just pulling out a few of those interactions from that big tree that you had in the background, now looking at this particular Cullen protein. So Cullen proteins are also known as what kind? E1s, E2s, or E3s? E3 proteins. So this E3 protein interacts with all of these other proteins that it's connected with in a line. So not surprisingly, these, and many of them, all those that have dark names, are actually known to be modified by ubiquitination. Not a big surprise. Interestingly, however, these white ones were not previously known to be modified by ubiquitination. People just found that there was a protein-protein interaction between these two. And we'll get into the methods right towards the end of the class. But pretty good indication that these are going to be modified through ubiquitination. And there'll be some process, either signaling or degradation, which is to be regulated through these particular proteins. So looking at all of these, you can get a bit of an idea how they interact with each other. And in many cases, things that interact with each other are all involved in one particular aspect of how the cell is working. So here, for instance, the origin of replication. There are lots of proteins that are involved in the origin of replication, which also interact with each other. Cell cycle regulation, all these guys interact with each other, again, not terribly surprisingly. But it's a way of just, if you look at protein-protein interactions, you can get an idea of, particularly, we get to sort of the genome part of this talk at the end, or maybe on Wednesday, getting an idea how things work. Because we can get sequences really easily. In fact, the whole human genome sequence, a bunch of other sequences, we've got all those things. But the question is, how do they work? And one of the ways to try and figure out how they work is to figure out who they're associated with, guilt by association. Okay, so that's it for proteins. Again, it's more review from last time. We talked about the amino acids and peptide bonds, secondary structure, hopefully beating the dead horse here. Protein folding is how those secondary structures all come together. We'll talk a little bit more about how that works later on when we talk about translation. Look at some of the structures here. Domains are these separate pieces of the protein which fold up, have their own structure by themselves. You, of course, can have their own function. But a little bit about multimers. Multimers are really important for having genetic minima. So you've got multiple copies of the same protein that then come together, and it's coming together which is really important for their activity. Talk a little bit about protein families. Again, it's all about the structure. Didn't really talk too much about modeling. Allostery is just those interactions far away from another part of the protein. So an interaction at one part of a protein causes a change in the structure in another part of the protein. And then those structural changes that we talked about mostly having to do with phosphorylation and G proteins, but also just adding ubiquitin. That's going to give you a structural change as well. So any questions about protein before we start talking about DNA? You'll see me for the next couple of weeks carrying this model around. One of the reasons I love this model so much is there are at least 4 problems with it. Probably 5 or 6. But come down and take a look at it afterwards. I think it's a really good review. Actually interesting, the prep lab put it together wrong, and it turns out it's really cool because they put it together wrong. So start out talking a little bit about the history of nucleic acids, and then, again, hopefully all of this is review. A's, T's, C's, and G's. What are the building blocks? We talked about helices before. Basically, these are building blocks that fit together in exactly the same way above and below them. And so that way you can have a nice repeating structure. Something like a helix, for instance. Chargaff's rule in 51. No, I don't expect you to know dates. But there are a couple of important things, I think, anyway, as far as is concerned. One is that a lot of the people involved here are still alive, which is really kind of cool. The second thing is that my parents were actually in Cambridge when some of these experiments were happening. So this is very, very, very recent. So when I mentioned last time, the 5th edition and the 6th edition of the book, 6, 8 years later, it's not been very long. This course didn't even exist more than about, a little over 50 years ago. Maybe 60 now. I'm getting old too. Sorry, back up one. So bacterial transformation, this was how people have originally found out that DNA was a genetic material. People thought the protein was a genetic material. Actually, for very many years, it was pretty clear that protein was a genetic material because DNA was just too big and too dull and too boring to worry about. Eventually, Hershey and Chase published their experiment in 52, and the double helix was published in 53. Now, so this is basically a period of 51 to 53, about 3 years, which is just amazing that people have gone from analyzing the individual nucleotides to ending up with a model for the structure of DNA. So a little of the history. The original discoverer of DNA, at least the one who is credited with discovering DNA, is Friedrich Michno. In 1871 in Basel in Switzerland, was an MD, just basically looking at what he could find in cells. Originally white blood cells, but couldn't get enough of this stuff from them. Looked at salmon sperm. We still use salmon sperm in labs today as a source of DNA because it's just loaded. One of the nice things about sperm is it's basically just DNA. It's a little bit of protein, but it's almost all DNA. And what he found was it was big, had a lot of phosphorus, and was acidic. So that's where nucleic acid came from. Nucleotides, that was the original discovery. What do you have in white blood cells and salmon sperm? That's all that it is. People then later, basically through, well, it's the rediscovery of the microscope, noticed that chromosomes were in cells, and those chromosomes seemed to divide, and it seemed to be that chromosomes were really the genetic material. So, okay, what's in the chromosome? Well, it turns out that there's a lot of nucleic acid in chromosomes, but there's also a lot more of these interesting proteins. Proteins got 20 different side chains, lots of diversity. Certainly something you think about complexity, that's much more likely to have the genetic material in it. Turns out also if you look at chromatin and chromosomes, most of the weight actually is protein. The DNA is actually relatively small in terms of amounts that you have there. Probably the main person who was involved in telling people that DNA wasn't the genetic material, was Phoebus Levene, 29, which is also like about 20 years before this had this big switch. He actually found out that it was a 2', in fact, deoxyribose is the sugar in nucleic acid, but you have only 4 of them, which is really kind of boring. So instead of having 20 amino acids, which is really cool, you got a whole bunch of diversity, it's likely to be something which is going to cause changes between what a salmon looks like, what a white blood cell looks like, et cetera. But you've only got 4 nucleotides. It's pretty dull. Now, why did he not figure out that this was more likely to be the case? Did anyone know about Phoebus Levene's experiments? You read about them? Basically, he took dog enzymes and broke down DNA with them into individual nucleotides, and then looked at those individual nucleotides and found out that there were only 4 of them, and found out also, again, this is a 2' deoxy sugar. So why was this such a big question? Again, only 4 nucleotides. Turns out that since he was using dogs and humans, there are very similar amounts of the C's, A's, T's, and G's. All 4 of them were very similar in terms of their amounts. And you'd expect that if you have something which is genetic material, it has a lot of diversity in it, that you'd have a lot more differences relative to each other. And so the prediction was, it was just a repeating polymer. DNA was a scaffold. DNA was a scaffold that held the proteins together in the chromosomes. So this is what everybody believed, or almost everybody believed, for about 20 or 30 years. So how did that start to change? Well, first of it was transformation. This is, in fact, not the figure from the text, but I think it's a little bit nicer one. In about the same time, these bacteriologists, and who cared about bacteria? Ugh, boring. Had discovered this effect, which was really kind of bizarre. And they couldn't quite figure out what it was. There was the S-strain virulent bacteria that if you inject it into a mouse, the poor mouse dies. On the other hand, a very similar kind of bacteria, and in fact, you could even get mutations in this kind of bacteria that became this kind of bacteria, that now you inject this bacteria into a mouse, the mouse is perfectly happy. Okay, that's fine. It's not too big a deal. But what's really bizarre is that you can heat up this S bacteria and kill it, inject the mouse is perfectly happy. However, if you mix together this dead bacteria with live bacteria that doesn't otherwise kill the mouse, you kill the mouse. So this was the transforming principle. So all it was doing was transforming this non-virulent bacteria into virulent bacteria. So how the heck can you do that? It's a pretty cool trick, actually, if you can in fact do it. So this was found, again, almost exactly at the same time they found that deoxyribose was the sugar that was involved in putting together DNA. Nobody really believed that it was important. Bizarre things that bacteria do. Who cares about bacteria? Well, about, again, 20-odd years later, there was a group at the Rockefeller in New York which thought this actually was really kind of cool. Let's see if we can follow it up a little bit. Let's see if we can figure out what that particular transforming material is. How do you figure out what it is? It took them a long time to do this. Again, 1944 was when this is published. This is, in fact, a figure from that paper in 1944. What they found was by mixing together heat-killed cells that look like this. These are the smooth cells, the S. These are the rough cells, that they could transform these guys into these guys just by adding something that they purified from here. And that something that they purified was active when they put in RNAs, active when they put in protease, but not active when they put in DNAs. And that was basically their experiment. They purify something, you treat it with DNase, that activity goes away. They treat it with RNase, treat it with protease, it doesn't go away. Basically, nobody really believed this at the time.
and nobody could care. There's some great stories about this Avery going to a meeting, scientific meeting, and talking about his new discovery about the transforming principle. And he said no one could hear him because everybody's getting up because they'd just heard the Dramamine talk, which was really cool, and they're all going somewhere else. So just like here, after I ask the click questions, everybody leaves. So what it turns out, of course, is that it is DNA, and what happens is basically shown here. You have your smooth cell, which encodes the capsule gene. And so that capsule gene, that's what gives you this slimy stuff on the outside. If you isolate the DNA, that capsule gene, you put the two of them together, and this was basically luck, because most cells don't pick up DNA from the environment. But this particular strain does. They called it pneumococcus at the time. Now it's a pseudomonas strain, I think. But the pneumococcus then picked up this extra gene, brought it into the cell, recombined it into the cell, and we'll talk about recombination later on. And then now this cell expresses the capsule, and that also makes it infectious. So transforming material is DNA, or at least it's not degraded by RNA and not degraded by protease. Again, basically nobody believed them. Yeah?
No, I think you answered it. DNA degrades it, so it makes it not have.
Correct. DNA's treatment gets rid of the transforming activity. RNA's treatment leaves it just fine. Protease treatment leaves it just fine. So, and if you're interested, I have copies of this paper too. It's actually kind of a fun read. About the same time, Edwin Chargaff, who basically told Watson and Crick that they were full of it when he met with them, was studying the actual amounts of adenine, thymine, cytosine, and guanine in DNA and getting much more accurate measurements than Levine had ever done. But much more importantly is what Chargaff did is that he looked at much more different organisms. Levine basically looked at dogs and vertebrates, which all have pretty similar amounts of C's, A's, T's, and G's, certainly given the analytical techniques that they had at the time. Chargaff was a really good biochemist. He's very good at quantifying the individual nucleotides, but much more importantly, he picked bacteria to look at, and particularly bacteria that had very high, what we call GC content, so guanines and cytosines. And so what he saw was in the tubercle bacteria, which is actually Mycobacterium tuberculosis, which has a very different concentration of G's and C's than it does of A's and T's, and very different relative to all the other organisms that people have been looking at before. Again, most of the vertebrates, the salmon sperm, the white blood cells, etc. So because of that, it's not a nice repeating structure. It's not just CATG, 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 CATG. So that was the big thing, was it's not a repeating polymer. He noticed that the concentration of A's was about the same, and actually I should put the squiggly lines here as T, and Z was about the same as G. I've tried to find the actual reference to Chargaff's rule. There's no actually really good reference about this. It's mentioned in a whole bunch of different places, but nobody actually, there's no publication of the rule per se. And it was, again, just that, you know, A's were about the same as T's, and G's were about the same as C's. And in one of his lectures, he says, oh, there may be an important thing about this, but I'm not quite sure what it is. And again, this is in 49, so very soon before Watson and Crick come along. Even more soon before Watson and Crick come along are Hershey and Chase's experiments. Hershey and Chase decided that, no, bacteria, they're really cool, but viruses are even cooler. Completely agree with them. Looking at bacteriophage. Now, why bacteriophage? Why were they so interested in bacteriophage? Bacteriophage are a really good thing to try and answer a pretty fundamental question, and that is protein versus nucleic acid. Why is that? Because these bacterial viruses are just made of nucleic acid and protein. There's none of this annoying lipid and all of the other stuff which is there. And particularly the case for these the so-called T-even bacteria. And the reason they were called T-even is because a bunch of bacterial virus researchers, also called the phage group, got together and said, this one is T1, this is T2, this is T3, this is T4. Just decided. And those T-even bacteriophage had just DNA and protein. It was quite a lot of DNA and quite a lot of protein. They're also really big. The electron microscope was also just being developed at this time, so they could see these particles on the outside of the cell, but they knew the only way that these viruses could replicate was to get their genetic material inside a cell. So, what did they do? Al Hershey hired an extremely good technician, Martha Chase, to do some of this work. This is the Cold Spring Harbor Laboratory Labs on Long Island outside of New York. Their bell tower, when you go there, has a C, A, T, and G at the 4 directions instead of north, south, east, and west. And so they did, particularly when I say they, it's really Martha Chase who did these experiments, where they looked at trying to piece apart this problem. So they had the electron microscope was a technology development. The other technology, which came along at about this time, was the availability of radioactivity. Why? Because there was a Manhattan Project, and people dropped a bomb, 2 bombs in fact, on Japan. And those people would be us. And the US dropped. But that meant that there have been huge amounts of development in looking at radioactivity and the production of various different radioactive isotopes. Particularly here, radio-labeled phosphorus and radio-labeled sulfur. Why phosphorus and sulfur? Well, we look at the DNA structure, there's a whole bunch of phosphorus in it, and that was actually what Friedrich Michener showed. There was a whole bunch of phosphorus in DNA. Proteins, there's actually some phosphorus in proteins because of the kinases that we talked about before, but there's no sulfur in DNA. So if you look at these 2 different radioisotopes, then you can separate the radioisotope that you have in DNA from the radioisotope that you have in protein. Your bacteriophage are made of DNA and protein. So you grow your bacteriophage with radioactive phosphorus, so you end up with bacteriophage that have radio-labeled DNA. You grow them with radio-labeled sulfur, then they have proteins that are labeled with radioactivity. Separate them from the cells. This is the famous blender experiment, the E. coli daiquiri, if you like it that way, where you can separate those viruses, which are on the outside of the cell, these guys, from whatever's on the inside. And it was that blendor experiment, actually, as they called it, which was really critical in terms of making the separation and also centrifugation. And then you just look, where's the radioactivity? Is it in the cells? You have your P32, indicative of DNA, or your S35 when it gets passed along. It's just a cartoon version of this. Again, they're actually 2 different experiments. You don't have S35 labeled and P32 labeled at the same time. You do 2 experiments. One where you have S35 labeled. You look and see, is there any S35 left at the end? One where you have P32. You have an infection. You remove what's on the outside. You let this replicate. You look and you see what radioactivity do you have here. This is the P32 experiment, the S35 experiment. And then in some cases, what's the new bacteriophage? And in this case, had P32 that was associated with it. So, clear to everybody? Yes, we're happy? Clear enough you can answer a clicker question on it? Good. I'll give you a second to fire them up. I need to start mine as well. If Chase and Hershey had found that the S35 was passed from an old virus to new viruses in their experiment, they would have concluded that DNA was the genetic material, RNA was the genetic material, protein was the genetic material, they had a contamination, or the viruses contain protein. again, feel free to discuss this. Everybody should get this right, I hope. Because you all nodded. You all know it. Stedman loves these kinds of questions, flipping over experimental results to something else. Okay, we've got 109 of you. I think that's as many as voted before, so we're getting no, 112? Where do those new people come from? New clickers. Oh, viruses, exactly. Yes, so they would have concluded the protein was the genetic material because they were looking for that particular radioactivity. Nope. Why does this thing kills me every time? Okay, that's it for clicker questions. You can pack them away now. So with 52, when this was published, Watson and Crick here in Cambridge, so slightly before my parents got there, but about the same kind of time. This crazy American guy who just finished up his PhD, I think in Illinois or Wisconsin, decided to come to England and work together with his PhD student, who was actually older than he was at the time. And I should have the arrows here. This is Crick. That's Watson. What did they know? Well, basically, they knew the structures of the nucleotides, and the structure of the nucleotides was really from the 20s. You know, Phoebus Levine, looking at the 2'-deoxyribose and looking at the various structures, the purines and the pyrimidines. So it's actually been really known quite a long time what those nucleotide structures were. That we had 5' to 3' phosphodiester bonds was a relatively new thing, which they'd known. And then they knew from, in fact, hearing a talk by Chargaff, and there's a nice book. Anyone read The Double Helix, The Search for the Double Helix, Watson's book? It's actually really quite short. It's a good read. I can loan it to people. I've got it in my office. I think they've got it in the library as well. It's pretty short. Chargaff came and gave a seminar, in fact, in London. They went down to London, and they heard him talk, and he came and talked to them and said, oh, you guys are completely full of it. You don't have a clue. But they knew that C's and G's were about the same concentration. A's and T's were about the same concentration. And then in the book, they talk a little bit, or should I say, Watson talks a little bit about stealing Rosalind Franklin's data from fiber X-ray diffraction of DNA. And so this was a really very new technique, and basically what Rosalind Franklin and her, at least in theory, supervisor Maurice Wilkins had done was take pieces of DNA, and DNA from regular sources like salmon sperm, and basically stretch it out and shine X-rays down it. And then those X-rays were scattered depending on where the individual nuclei are in whatever you're looking at. So what do these patterns look like? They look like this, and at first you go, how the heck can you get anything out of this data? And how can Watson and Crick, just looking at these data, get an idea for their particular structure? Well, it turns out that what Crick was working on at the time was diffraction of protein structures, particularly containing large numbers of helices. The lab that they were in was the famous one that was working on protein structures. So protein structures were known way before any of the nucleic acid structures were known. And so Crick was really kind of primed for looking at these kinds of structures. And basically, what they were able to get from these kinds of structures was that it's about 2 nanometers across, clearly helical. That's this X structure. The 2 nanometers is from the top to the bottom. And then most importantly were 2 repeating structures. There was a repeating structure at 0.34 nanometers and 3.4 nanometers. So these are the data that they got from Rosalind Franklin. Of course, unfortunately, Rosalind Franklin died before the Nobel Prize was given out for this. They don't give Nobel Prizes posthumously, so she wasn't cheated out of the Nobel Prize. They don't give Nobel Prizes, unfortunately, to people who've passed away. The description of Rosalind Franklin in Watson's book is not very positive, but later on in life, they actually did end up working together quite a lot before she, in fact, died. The main reason she died was a leukemia, probably due to the radiation from the X-rays, unfortunately. They also, and this is actually shown really quite, or discussed quite nicely in the book, they had a model that Linus Pauling had been putting together in terms of what DNA should look like. He'd also had a helical model because he was the one who came up with the models for what? Proteins. What kind of things in proteins? So alpha helix and beta sheet, both of those came from Linus Pauling. So the fact that Watson and Crick, these two upstart British-American team, ended up beating the man from Portland was really kind of a surprise. So again, overview of what they knew. They knew that these were 2'-deoxyribose. I want to go through this structure in a little bit more detail. We didn't talk about it so much when we talked about the chemistry, because I'm going to talk a lot about 2' and 3' having to do with the polarity of DNA molecules. Remember, we got starts and stops, beginnings and ends. So all the primes have to do with the sugar. So 1', that's where the base is attached. 2', deoxy, so you only have hydrogens here. 3', OH, that's one end of your molecule. 4', which is the next carbon. 5', where you have this connection usually to the phosphate. We already talked about this when we talked about ATP. ATP has 3 phosphates hooked on here at the 5'. In the case of DNA, in the DNA molecule, there's only 1 phosphate, which is attached here. This just shows the reaction of adding the base to your ribose present right here. 2 flavors of bases, got big ones and little ones. The way I remember this is that the big molecules have the short name, the purines, and the small molecules have the long name, the pyrimidines. So this is what they knew. They knew that the attachment was here 5' to 3' between any of the individual nucleotides all put together. It's a little bit easier way to look at this structure. Again, they got the helix because there was this crossing over structure, and all of these repeats based on where the lines were, and then they had to make a model to fit these data. And so that's exactly what they did, and this is the only figure from their paper. And rumor has it, it was actually drawn by Crick's wife. So we didn't have the wonderful PowerPoint and everything else these days. All of these things were completely hand-drawn. So now, again, this is the 5' to 3' linkages here. One end, 5' end of your chain hooked up to the 5' carbon in your sugar. Here's the 3', here's your next phosphate, it's just the 5', etc. All of these all the way down. This is just a repeat of these panels which are present in the book. So what they get, basically, this one paragraph is pretty much the whole paper. They built models. That's all they did. They're really much more theoretical than anything else here. They didn't do any experiments. They built models. And they built models and put them together so that they fit the data. So it was all a complete model building. So what they came up with were these 2 complementary strands, and complementary due to the base pairing between the 2 of them, that wrap around each other in a pair of anti-parallel helices. I'll get to you in just a second. The anti-parallel is that the polarity now of the strands is different. So you have to have a 5' end that pairs with the 3' end at one end, and then the 3' pairing with the 5' at the other end. This is the only way you can get this structure to work, is you have them anti-parallel to each other. Right-handed helix. This is also a nice right-armed helix here. You take a look at this again after class. Take your right arm and wind it through. It actually goes nicely into the DNA. I try and do this with my left arm. It just bangs. So I've also got a few more of my helices here to take a look at. Phosphates on the outside. This was the big difference to Pauling structure. He had the phosphates on the inside. How you can get phosphates on the inside of a structure is really bizarre because, of course, they're going to be repelling each other. Got a nice stable negatively charged. And the bases paired with hydrogen bonds in the middle of the helix perpendicular to the strands that are present on the outside. How do you get this to work? You've got to play around a little bit with the structures of the chemical natures of the bases. You have to be amino groups in cytosine and adenine and keto groups and not enols in order to get this base pairing to occur. Through that process, hopefully you can see it here, when you build the model, it's not symmetric. If it were completely symmetric, it would be right around this bar in the middle. It's not the case. It's offset a little bit. And that offset business means that you end up with a asymmetric sets of potential places to interact with the DNA. You have a major groove and a minor groove. Major groove, I'll get this pointer here, is here. It's this space right here, this major groove of DNA, and here's the minor groove of DNA. Major groove, and again, the model here, you can see it, got a major groove and a minor groove right next to each other. So there's a question.
Yeah, the 0.34 nanometers is
Yeah.
the distance between the two strands.
Okay, so the question is, what about those repeats? So the 0.34 and the 3.4. It turns out that those repeats are a turn. So from where this backbone molecule lines up on top of the next backbone molecule. And so that's one of your repeats, because that repeats again and again and again. And then the other repeats are between the bases. So what does that mean in terms of how many bases you get between one turn of your helix? 10. Very convenient. Okay, and then this was published 25th of April, 1953. It's on D2L. Read the paper. There will be a question on the exam. Just one. So, right-handed helix, again, hopefully kind of beating on things here. The other aspect has to do with, again, what kinds of, we have all of our keto groups here on cytosine, keto group here, amino group here, amino group here. And what that means is you have these hydrogen bond donors and acceptors which can form across the middle of the double helix. This is the model. Anyone recognize this? Old enough to know what this is a reference to? It's only a model, which is true. It is only a model. Turns out that the real DNA structure doesn't look like that. What we're going to talk about basically for the next couple of weeks is that real DNA doesn't actually look like that. But the model itself, this is a picture of that model. They have one, I think there are like 3 copies of this model. One of them is the Smithsonian. It's actually really pretty cool. It's big. It's like this size, this big around. And there's also some pictures of Watson and Crick pointing at it. And the main thing that they had to wait for was the machine shop at Cambridge actually making each of these individual nucleotides to take a look at it. One of the mistakes that Watson and Crick made is they actually thought that both of these, the GC and the AT, had 3 hydrogen bonds relative to each other. They thought it was actually a hydrogen bond between these two. This is actually incorrect as far as that was concerned. These are the Watson and Crick base pairs between A's, T's, and G's and C's. With these hydrogen bonds form, you remember hydrogen bonds have to form in a more or less linear interaction between the hydrogen bond donor and the hydrogen bond acceptor. I always ask people to memorize base pairs. You may have seen that on some of the old exams. You go back and look at them and say, draw a base pair. This is actually missing a couple of things as far as a base pair is concerned. It doesn't have the hydrogens. There should be a hydrogen here. There should be a hydrogen over here. It also doesn't have sugars. But the main thing that you don't see on here is that the polarity of these two strands is different. Here, it really looks very much like they're the same. So what you need to do, if you're going to draw base pairs, which again I'm not going to expect you to do, but there'll be something about base pairs on the exam. Here, 3' going down to 5', and 3' going down up to 5'. So these are always going to be anti-parallels. The only way you can get these things together. And that, to me, you know, above and beyond the hydrogen bonds, most people think that was the huge breakthrough that Watson and Crick made, was the hydrogen bonds in the middle. I think just as important was the fact that they had to be anti-parallel relative to each other. Because that's something they couldn't get from the X-ray data. They knew they could get an idea that the phosphates were on the outside. It was more of a guess. But they knew what these repeats were. They could sort of guess where you were getting interactions, and you could model that pretty easily. But modeling 5' and 3', you know, you just had to basically play around with the models and figure out the one had to be back to front relative to the other one. So this is when I keep beating on polarity, 5' versus 3', and always anti-parallel relative to each other. Here's a nice example of the major and minor grooves. It's a space-filling version of DNA. Here, space-filling again, this is the van der Waals radii. Here are all the phosphates on the outside. Here's your major groove. Here's your minor groove of DNA. So another way of looking at it here again, major groove of DNA, minor groove of DNA. Here, this again emphasizing the different polarities. Here, going from 5' to 3' on the red strand, and from 5' to 3' on the blue strand. They're opposite polarities relative to each other. Some of you may have noticed the A-form versus B-form DNA. In fact, those two images that I showed when we first talked about Rosalind Franklin's data, she actually had two kinds of DNA that she was looking at. The best data was actually from this particular form of DNA over here, A-form DNA, which also is same kind of right-handed helix, both anti-parallel relative to each other, but much more compacted. It's really hard to see much in the way of a major groove or a minor groove here, and really overwound relative to each other. And the reason that these are so different from each other, this particular one, the fibers here had a lot of water that was associated with them. Here, very little water that's associated with them, so much more compacted. And the main reason that I show this is that it was only a model. This is the way that some DNA looks some of the time. This is the way that some DNA looks some of the time as well. This is just as good as this in terms of representing the DNA. It's just that this one's a lot easier to build a model for. This one's a heck of a lot harder to build a model for. So why do most people look at this structure? Why am I beating on this major and minor groove so much? One of the reasons is that if you're a protein and you want to figure out where to bind to on your DNA, you need to be able to figure out what that particular DNA sequence is. How do you figure that out? Well, it turns out that most of the chemicals that tell you that there's a difference between an AT or a GC base pair, even when they're hydrogen bonded to each other, are present in the major groove of DNA. So if you look here, this is just the 4 different base pairs, CG, GC, TA, and AT. All of these have different atoms that are present in the major groove. So 2 hydrogens here, hydrogen that's part of a hydrogen bonded intermediate. So this one's going to be slightly different. It's going to have a slight positive charge on it. Here's your hydrogen bond, an O here. Here you've got a methyl group that's sticking in the major groove. One really easy way to find major versus minor grooves. Look where the methyl groups are from thymidine. Thymidine always has a methyl group that's hanging out on, and it's going to be in the major groove of DNA. Also true down here. And again, it's just this pattern of hydrogen bond donors. Say, donors should be these blue ones. Is there an earthquake? Hydrogen bond donors versus hydrogen bond acceptors, and methyl groups that are all going to be present in the major groove of DNA. The minor groove, which is smaller, also has less chemical information. So if you're going to have non-specific interactions with DNA, it's very often going to be happening in the minor groove. Specific interactions are going to be happening in the major groove. And that's outlined here. Basically, if you look at where your hydrogen bond acceptors and donors are, in the major groove, you can tell whether you've got a GC, an AT, a CG, or a TA base pair. Or in the minor groove, you can't tell the difference between GC and CG, or AT and TA. Much less information here than you have here. Not surprisingly, over 4-odd billion years of evolution, turns out that the major groove is almost exactly the right size for an alpha helix, which has all of its amino acid side chains where? Sticking out. What are those now going to interact with? These molecules in the major groove of DNA. So it's not terribly surprising that these two are the case. I think we'll stop here. We'll talk about hybridization next time.